Envision a future where aging is no longer synonymous with declining health and vitality. A world where age-related ailments are mere relics of the past, and individuals can relish their golden years with the same energy and zest as their youth. This isn't a far-fetched fantasy, but a potential reality on the horizon. Dr. David Sinclair and his team at Harvard Medical School are at the forefront of a scientific revolution, aiming to reverse the aging process at the cellular level. This isn't just another fleeting anti-aging trend or baseless claim. It's a beacon of hope, a testament to the power of cutting-edge science that could reshape the course of human history. So prepare to be amazed as we delve into the awe-inspiring realm of Dr. Sinclair's research. To fully grasp the magnitude of Dr. Sinclair's work, we must go back to where it all began, with the discovery of the Yamanaka factors in 2006. These 4 proteins, OCT4, SOX2, KLF4, and C-mice, OSKM, were identified by Dr. Shinya Yamanaka as the key to reprogramming adult cells back into a youthful, embryonic-like state known as induced pluripotent stem cells, iPSCs. Induced pluripotent stem cells are those that are reprogrammed back into a state where they can develop into any cell in the body, much like embryonic stem cells. This revolutionary finding showed that the aging process could be reversed at the cellular level. It was like finding the Rosetta Stone of rejuvenation, a way to decipher the complex language of cellular aging and turn back the clock. However, there was a catch. While the Yamanaka factors could indeed reverse aging, they also cause the cells to lose their specialized functions and revert to state of pluripotency. In other words, a skin cell reprogrammed with the Yamanaka factors would forget how to be a skin cell and instead become a blank slate capable of turning into any cell in the body. This was a problem because we didn't want to turn all of our cells into iPSCs. We just wanted to make them young again while retaining their identity and function. This is where Dr. Sinclair and his team come in. They realized that the solution to this problem was to find the just right level of exposure to the Yamanaka factors. Not too little, not too much, but the perfect amount to rejuvenate cells without losing their identity. They coined this approach partial cellular reprogramming, and it's been shown to have remarkable effects in animal models. In mice, for example, partial cellular reprogramming has improved the function of aged tissues, including the brain, kidney, and muscle. In some studies, it's even been shown to extend lifespan. And the best part, the treated cells retain their specialized functions so they do not risk turning into iPSCs or causing tumors. But there was still a problem. Delivering the genetic material containing the Yamanaka factors to cells in the body is challenging. It typically involves the use of viral vectors, which are modified viruses used to deliver genetic material into cells. However, using viral vectors can cause complications and raise safety concerns. This is where Dr. Sinclair's latest research comes in. The hunt for small molecules that can mimic the effects of the Yamanaka factors without the need for gene therapy. Dr. Sinclair's team embarked on a rigorous and ambitious project, meticulously screening thousands of small molecules in search of compounds that could rejuvenate cells like the Yamanaka factors without genetic manipulation. They developed a high-throughput screening method called the nucleocytoplasmic compartmentalization (NCC) assay, which allowed them to swiftly and effectively test the effects of different compounds on cellular aging. This methodical approach ensures that only the most promising candidates for rejuvenation are identified, instilling confidence in the scientific process. The NCC assay is based on a fundamental principle of cellular biology: the separation of the nucleus and the cytoplasm. The nucleus is the cell compartment containing the genetic material, DNA. At the same time, the cytoplasm is the gel-like substance that surrounds the nucleus and contains various organelles and molecules necessary for cell function. In young, healthy cells, there is a tight control over what goes in and out of the nucleus. Specific proteins are meant to stay in the nucleus, while others are supposed to remain in the cytoplasm. However, as cells age, this tight control starts to break down. Proteins that should be in the nucleus start to leak out, and vice versa. This breakdown of nucleocytoplasmic compartmentalization is a hallmark of cellular aging. The NCC assay uses this phenomenon by tagging nuclear proteins with one fluorescent color and cytoplasmic proteins with another. This allows researchers to visualize the breakdown of NCC in real time and use it to measure cellular aging. The assay proved a powerful tool for distinguishing between young, old, and senescent cells. Senescent cells are cells that have stopped dividing and entered a state of permanent growth arrest, often in response to stress or damage. The NCC assay allowed Dr. Sinclair's team to screen thousands of compounds to find the most promising candidates for rejuvenation. After screening a library of over 80 small molecule cocktails, Dr. Sinclair's team identified 6 that showed the most promise for reversing cellular aging. These cocktails, dubbed the Magnificent Six, were able to significantly reduce the transcriptomic age of senescent cells. Transcriptomic age refers to the biological age of a cell as determined by gene expression patterns, which can be measured by analyzing the levels of RNA transcripts in the cell. Some Magnificent Six cocktails showed an impressive reduction of over 3 years in transcriptomic age after just 4 days of treatment. But the real kicker, the treated cells showed no signs of losing their identity or becoming iPSCs. They didn't express any pluripotency genes or markers of other cell types. In other words, the Magnificent Six were able to rejuvenate cells while preserving their specialized functions, the holy grail of rejuvenation research. The potential of these small molecule cocktails to combat cellular aging is monumental. If they can be developed into a safe and effective treatment, it could herald a world where age-related diseases are eradicated. Imagine a future where a simple pill could rejuvenate your cells without the risks associated with gene therapy. Dr. Sinclair's research is just the beginning of what could be a new era in aging research. With the discovery of the Magnificent Six, we're one step closer to a world where aging is no longer a one-way street. Of course, there's still a lot of work to be done. The safety and efficacy of these small molecule cocktails need to be rigorously tested in animal models and eventually in human clinical trials. In the future, we could see a world where people live longer, healthier lives and are free from age-related diseases. A world where 80 is the new 50 and where people can enjoy their retirement years with the energy and vitality of their youth. It's a future that many of us have dreamed of, but never thought possible until now. Of course, with any great discovery comes great responsibility. As we progress with this research, we must also consider the ethical implications of extending human lifespan. How will this impact society, the economy, and the environment? We as a society will need to grapple with these questions as we enter this brave new world of rejuvenation research. Dr. David Sinclair's research on reversing cellular aging with small molecules is a game-changer in aging research. By developing the NCC assay and identifying the Magnificent Six, his team has brought us closer to a world where aging is no longer a fact of life. The potential implications of this research are staggering. A world without age-related diseases where people can live longer, healthier, and more fulfilling lives. But this is just the beginning. As we unravel the mysteries of aging and develop new therapies to combat it, we must also consider the broader implications for society. How will we adapt to a world where people live longer? How will we ensure that everyone has access to these life-extending technologies? These are the questions that we must grapple with as we move forward. But one thing is clear. The future of aging research is bright, and Dr. Sinclair and his team are leading the charge. So the next time someone tells you that aging is inevitable, remember, a group of brilliant scientists at Harvard are working tirelessly to prove them wrong. And who knows, maybe one day we'll all be able to take a sip from the fountain of youth and experience the joys of life anew. Wouldn't that be something?